and welcome to Pitt Street Research. My name is Stuart Roberts, and I'm one of the founders of our firm. And with me is Dr. Tim Oldham, who's the CEO of AdAlta, ASX code A1AD. Tim, good morning to you.
Hi, Stuart. Great to be here.
It's let me just check the date here. It's the 20th of July. You've just announced, Tim, something very interesting for your AD214 program, which has basically delivered in spades in terms of what you've wanted to achieve in lung conditions. You're now going to turn it into a soluble, a nebulized form of the drug, so you can deliver it by inhalation rather than usual intravenous delivery. Talk to us about that program and what motivated the decision.
Yeah, we announced yesterday that we've elected to translate the work we've done with AD214 into an inhaled formulation for direct lung delivery, and that we expect to take that inhaled formulation through into the next patient clinical trials. What's motivated this? Well, I think I've always believed there had to be a more convenient way to deliver AD214 to patients in an intravenous injection. IV is convenient if you're in hospital already, but for a chronic disease, it's preferable to have something a bit more convenient. So inhaled has always been on our radar screen.
Speed of delivery is always an issue with these things, and you can get it to the patients much faster through inhalation, right?
Well, that's right. So it's more convenient for the patient. It's easier. They can do it at home. They can do it themselves. We have much more flexibility with the dosing regime. We'd initially started developing an intravenous version of AD214 because that was what we believed was the fastest way to get initial safety data and then potentially initial efficacy data. And certainly it's proved its worth to get the initial safety data. So our phase 1 program, which will now conclude with an IV formulation, has demonstrated that the molecule itself is extremely safe. We've also been developing a radio-labeled version of AD214 to enable PET imaging. And this is a really powerful tool for visualizing the distribution of a drug within a person. And that program is delivered in spades earlier than we thought it would. We were expecting to get particularly interesting insights from that when we put it into patients. In our preclinical studies, it's actually demonstrated, one, that we've successfully developed the radio-labeled version, but two, that it also highlighted we were getting a significant proportion of our intravenous dose was going directly to the liver. We haven't seen any evidence of liver toxicity, and we actually know that the drug is not going to the hepatocytes, which is the cells in the liver that are responsible for metabolic activity. And so that reinforces the safety of this, but the reality is that half the injected dose is ending up somewhere where it's not going to be active. And so that prompted us to accelerate our thinking around direct lung delivery. And we've now found that we have a pathway to be able to do that for our next clinical studies rather than having to wait longer. So we're able to implement that inhaled delivery formulation without delaying the progress to the next clinical trial. And that was important, obviously, from the perspective of continuing to drive this molecule forward from an efficacy point of view and for partnering purposes as quickly as possible. But now we're able to do that with a much more convenient inhaled formulation for patients.
Right. Now, people who know the protein drug space well maybe will have some concerns given molecule size and how easy it is to turn this into a nebulized formulation. Presumably, you have some experts you're talking to who are confident they can create this new formulation.
Yes, we have. So historically, there have been, the original inhaled drugs were often dry powder formulations. These were much more complicated to manufacture and control. With the advent of smart nebulizers, these are devices, we're probably all familiar with them from when we were kids, with colds and flus, but smart nebulizers now are designed to be very gentle to large molecules. They're also got very good control over the aerosol particle size, which is important to determining distribution in the lungs. And essentially, we now know what aerosol size is required in order to get any product into any part of the lungs, larger for the upper airways, smaller particles for the lower airways. And the various consultants that we've spoken to are supremely confident that they can deliver an aerosol version of AD214 to the right areas of the lungs. With a formulation that'll be very similar to our intravenous formulation. So that technology seems to be working well. There are 4 drugs for IPF in development that are inhaled already, one of which is a biologic. IPF patients routinely inhale other drugs, salbutamol and steroids, for example, for symptom relief. And obviously there are inhaled drugs available for cystic fibrosis and other pulmonary diseases. There are at least 2 approved biologics that are administered by inhalation today. There are at least 19 others that have achieved clinical stage development, demonstrate the ability to deliver the drug into the lungs is for a large molecule is feasible. And the small size of our eye bodies is helpful here. Most of the proteins that have been delivered by inhalation to date are in that range of 15 to 80 kilodaltons. Our AD214 molecule is 72 kilodaltons, and the eye body itself is about 15. So actually this could set us up for inhaled delivery of all sorts of eye body enabled products.
Absolutely, and that's quite exciting, because there's a whole lot of lung diseases which you could potentially go after. Any ideas what the team would be thinking of in terms of additions to the pipeline once AD214 in IPF and ILD gets a move on?
So there are a number of other fibrotic diseases that are potentially amenable to a local administration route. Obviously, eye fibrosis is the most obvious. Yep. There are also some potential topical applications. We actually have some hypotheses around why AD214 is distributing to the liver. And it may be that we can formulate around that to produce an alternate intravenous formulation that would then open up a range of other fibrotic indications. We haven't thought too much about other pulmonary applications yet. Obviously, we're focused on fibrosis. Any other indication would potentially require a new molecule. But we are thinking hard about the next products in our pipeline. We're close to finalising the next 2 internal programs. They will both be G protein coupled receptors like CXCR4 that AD214 targets. One of those is likely to be another in the fibrosis space as well. So that may be applicable in lung fibrosis, but it may be applicable in other fibroses with systemic application as well. What we do know is that this phenomenon we've seen with AD214 and the liver distribution appears to be unique to AD214. So we've also labelled a couple of other eye bodies. We've studied those in mice and large animals as well, and they do not display this same liver distribution that AD214 does. So it appears to be very specific to this molecule. It's not something that's a feature of eye bodies generally, and that sort of bodes well for the diversity of applications of other parts of the pipeline.
Now, finally, one of your more interesting programs, which we spent some time in a research note for Pitt Street Research, involves radio labeling an enzyme called granzyme B, and you're working with GE Healthcare on that for a specialized imaging application. Talk to us a little bit about the potential upside from that program.
So we've got more and more excited about this one as we've worked with GE. So the idea here is that the immuno-oncology drugs, the checkpoint inhibitors, have revolutionized the way we treat cancer. And essentially what we're now doing, rather than trying to kill the cancer, is to reactivate the patient's own immune system to fight the cancer. And that's what the checkpoint inhibitor drugs do. Unfortunately, they only work long-term for a small number of patients, between 20 and 40%. And the challenge we have is that we can't work out which are the 20 or 40% until the tumor progresses, which is obviously too late.
Right.
So the potential for imaging of a biomarker or an indicator of whether you're getting a response to one of these checkpoint inhibitor drugs is enormous. And GE came to us with a challenge and said, look, we know that when the immune system is activated and an immune cell is attacking a target like cancer, it secretes an enzyme called granzyme B, which essentially is the poison, the killer enzyme that kills the target cell. So if we could image an increase in granzyme B when a checkpoint inhibitor has been administered in the vicinity of the tumor, that tells us that the checkpoint inhibitor may be working. If we don't see an increase in granzyme B, then the checkpoint inhibitor probably isn't working. So GE came to us with a problem that said, look, we think this is a great biomarker. We'd like to be able to image it. We need a way to target it. We can deliver the radioisotope, but we need something to target it. And they've selected us to work with them to develop eye bodies that target granzyme B, deliver their radio-labeled imaging agent so that we can image granzyme B in these patients. And by doing so, we should be able to get patients onto the right therapy faster. We can stop dosing them if they're not responding. And from a drug development point of view, you would now have a very powerful tool that would enable us to work out whether a new drug is more effective or not than another one much faster than is otherwise possible.
Right. And it's impressive to me as well, that the speed with which this program can move. Once we know that we can image granzyme B, there are patients in hospital on checkpoint inhibitor therapy all the time.
Right.
That you can then run a very fast clinical study with the help of GE, which is one of the largest companies in the world, right?
That's right. So we've completed the discovery process now. We've moved into preclinical development, which GE is leading. They've actually engaged us to continue working with them on manufacturing and some of the in vitro assays they need to do. So that means we continue to generate revenue from this program even ahead of those future milestones. But you're right, the program to develop an imaging agent is much faster than that of developing a therapeutic.
Yes.
And so it's a relatively short preclinical program. Then it would be a relatively short phase 1 safety, then a potentially a pivotal phase 3 clinical trial, and then you're into commercialization. But importantly, because it's such a powerful research tool, as soon as you've got clinical proof of concept, GE can start selling that product into other clinical trials earlier, even before they've got a formal marketing authorization for general use, and that's because the approvals go through the clinical trial route.
Right.
So this could generate revenue much faster than a traditional drug, and could generate revenue even before royalty revenue, even before it gets formally approved. So, really exciting in terms of speed, but we're really excited about the potential applications of this as well. And these products generate sales north of, potentially the largest PET imaging agents generate sales north of $400 million a year. So even a single-digit royalty on that is a meaningful royalty revenue stream for a company like AdAlta.
Well, not only that, checkpoint therapy and the cancer immunotherapy in the larger sphere is postulated by some to be $100 billion market in the foreseeable future. And just a small percentage of that as the most efficient diagnostic to select patients. That's got to be good for a little Melbourne-based company like yours, right?
Absolutely.
It won't be so little soon.
Well, that's the objective, right? So we're on track now to progress 2 more internal programs. We're working hard on another external collaboration to go alongside GE. So we're on track to have grown from a single product company, a couple of years ago to a 2-product company now and 5 products by the end of the year. So certainly that momentum is really starting to build for us, and we're really excited about the future coming ahead. And we're super excited that for AD214 we now have a really clear pathway to patient efficacy studies with a very clear and convenient patient formulation. And we can do that without any delays to getting that efficacy data. So, super exciting last couple of months for us.
Dr. Tim Oldham and team, well done. Keep up the good work.
Thanks, Stuart. Great to talk to you.